This is the standard operating procedure for the cross-flow setup of the membrane filtration experiment. This video will take you through the preparation, operation, and shutdown of this equipment for the experimental runs. To begin, we will go through each piece of the equipment, what it's called, and what it does. The hopper is the conical vessel on the left. This is where water source is initially placed before it pumps out the bottom of the hopper and circulated through the system. There are also tubes that enter the top of the hopper. One is from the end of the circulation that deposits the returning water. The other is from the bypass to control inflow rate. The pump is sitting behind the hopper. It moves the water from the hopper and through the tubes of the experimental system. The control panel to turn on the pump sits below the table next to the chiller. The tube from the hopper to the filtration chamber has a valve on it to control the flow and pressure gauge to read the water pressure. Before the valve, there is a tube line that can bypass water to return it to be deposited in the top of the hopper. By restricting flow using the valve, more water will flow through this recycled route. The water that does continue past the first valve will then run through the filtration chambers. There are 4 total, but only one will be mostly used for this experiment. From this perspective, it is the back left. The line leading to the front chambers will not be used and therefore will need to be properly isolated with this valve. The chamber in series before the one we will use should be isolated with a solid plastic card within the chamber. During the prep portion of this video, we will demonstrate how to change the cards within the chamber. After the filtration chambers, the water flows through tubing to be deposited in the top of the hopper. There is a valve to control the flow and a pressure gauge on this line, along with a flow meter. There is also permeate tubing that connects to the top of the filtration chambers. This will be used to measure volumetric flow rate of permeate or to collect filtered water sample. Next, we will discuss the preparation necessary before running an experiment. Now, the purpose of this experiment is to determine the flow rate of water moving through the system with various membranes placed inside the filtration chamber. So, to run the experiment, the membranes need to be prepared in advance. Let's take apart the filtration chamber to see what we'll need. To disassemble the chamber, use a wrench to loosen each of the 4 knobs and unscrew them. Then, carefully remove the top portion of the chamber. You can see here the very porous metal plating in the top portion. Sitting on the bottom half of the chamber, we can either have a plastic card or a membrane sheet. The plastic card prevents flow through metal plating and therefore the permeate tubing, so all of the flow is directed through the circulating system. The plastic card will be used when clearing the system with fresh water before and after an experimental run. The membrane is for the experiments themselves, when we are trying to measure the volumetric flow rate of water through the membrane. These cards come from a roll of membrane paper that can be found in the lab. Before the experiment, you need to cut out membrane cards using the plastic card as a template. Be very careful when cutting out the cards. Getting any sort of dirt or smudges on them compromises the membranes and they cannot be used for the experiment. Always wear gloves when cutting out membranes and when conducting this experiment. To ensure proper flow through the membrane cards during the experiment, they need to be soaked in DI water for a minimum of 1 day. Now, before running the experiment, the system needs to be thoroughly cleaned. This can be done by running the system as you would for the experiment, but with a plastic card instead of a membrane card, as we previously mentioned. This is so that all of the water is fully circulated and none exits through the permeate tubing. Additionally, if a part of the process is visibly dirty, such as the hopper or the inside of the filtration chamber, clean it before running the water through. The water should run for a minimum of 3 hours. Because the clearing method is the same as the experimental method, minus a few details, we will explain the experimental procedure in this video, but also note where the cleaning methods differ. Now, we will discuss the experimental procedure. When conducting a wastewater experiment, the proper card needs to be placed within the filtration chamber. To change the card, open up the chamber using the method previously described and place the membrane in the chamber with the smooth side down. Make sure that it is properly placed and aligned in the system. For the clearing run, you will use a plastic card. Then, reattach the top of the filtration system, tightening the knobs well so that there is no chance of leaking. Turn on the power for the chiller. The main power button is in the back side of the chiller. Using a 10-liter bucket, collect the wastewater. For the clearing method, collect the water from the sink in the back room. It is a high pressure system, so to collect water at a proper flow rate, slightly open the red valve and then slightly open the orange valve and fill the bucket. After ensuring that the valve on the bottom of the hopper is closed, pour the water into the hopper. The minimum quantity of water is 6 liters to operate the setup properly, but 10 liters is recommended. Next, create a proper flow path throughout the system. This is done by ensuring that the valve leading to the front 2 chambers is still closed and opening the valve leading to the back 2 chambers. To start the pump, press the green button in the control panel and speed it up until the display reaches 60. Once it is ramped up to 60, adjust the back and front valves that are next to the pressure gauges in tandem so that the back pressure gauge reads 10 bar and the flow meter reads 2 liters per minute. You will need to systematically adjust the 2 valves throughout the experiment to be sure they remain at 10 bars and 2 liters per minute. For the clearing run, use about 3 liters per minute with the back valve almost 100% closed and the front valve almost 100% open. This means close and open the valve all the way and then turn it a quarter turn in the opposite direction. The bar readings are written in red on the gauge, and the portion used to read the flow meter is the bottom of the mushroom cap, halfway down the floating piece within the device. Next, place the end of the permeate tubing in a graduated cylinder. The tubing will be slowly filled with filtered water. Once there's enough to exit the tubing and begin filling the graduated cylinder, start recording the time. Remember to keep an eye on the flow rate and pressure readings and to maintain them at the proper setting of 10 bar, 2 liters per minute. Once the time period is up, remove the tube from the cylinder and measure the volume. Divide the measured volume by the recorded time to calculate the volumetric flow rate. Once the experiment is completed, it's time for the cleanup. Use the pump control panel, reduce the motor speed to 0, and then press the power button on the panel to turn off the motor. But make sure to leave the pump plugged into the wall. Next, close the valve leading to the back filtration chambers. Take the bucket used to fill the hopper and place it underneath the valve at the bottom of the hopper. Open the valve and drain the hopper into the bucket, and then close the valve once empty. Pour the water down the sink. Next, open the filtration chamber. Replace the membrane with a plastic card to block the outflow of permeate. Then close the chamber. Repeat this general procedure with clean water to clean out the system, starting with the instructions to create a proper flow path. Once the system clearing is completed, follow the shutdown instructions previously described and turn off the chiller.